I had no idea what any of these were. She was like, that was like
okay, before we start tonight's exercise, which is working with our physiological unknown, we have to look at the results from last time's exercise, the determination of microbial numbers, and also the use of selective and differential media. But we're also going back to our food micro one more time. I want you to check the sauerkraut pH. So when we started, our pH was 6.5, 6.5, around in there. We need to check the pH and see to what extent it's dropped. The other thing that you're supposed to do tonight is gram stain from the cabbage surface. When you gram stain from the surface of the cabbage, you're not going to have very much cell material, so you kind of hold a little piece of cabbage and scrape it with your glue. You're going to scrape it onto a glass slide and make a little bit of a smear. It's going to be very light. So when you do the gram stain, don't decolorize very long, or you will over-decolorize. There's not going to be very many cells there. The other thing we're supposed to do is gram stain from your buttermilk yogurt plates. Now, we're going to be doing gram stains with the physiological unknowns as well. So I would like you to put all of these gram stains on the same piece of glass. Might get a little crowded. And if you can't do it, if you can't get all of them on there, I've done as many as 7 gram stains on a piece of glass. Because remember, your smear doesn't have to be big. If it was 1 millimeter across, that's still 1,000 cells. Probably. They're little, so you can make your smears really small. Anyway, we are supposed to gram stain from these, and then I want you to turn those plates in. This is kind of like turning in the soil plates. When you turn in the write-up of the soil enrichment exercise, you turn in the plates are worth 5 points. These plates are worth 5 points. So turn the plates in. And then you can sample your apple wine. You can sample the sauerkraut too if you want to. This is optional. However, what I'll do is set our food micro cart up out in the hallway. We'll take our apple wine samples out there, put them on ice. There'll be pipettes to transfer in some tiny little cups. You can sample the apple wine. And it might be pretty decent at about this point in time. I think that's all we're supposed to do. So the next thing we're looking at is determination of microbial numbers. For this, you're going to need your numbers plates. I put them on the front edge of the counter. So they're on the side counter over here, right on the front edge. You should have a stack of 3 plates. So if you would like to get your lab coats and get your numbers plates, that would be helpful. Yeah, so NJ.
I think I actually have some in my bag. It's so decent now. Remember that time you finally because I whispered it? No. You need some Lab, girl.
I'm sorry.
Well, I knew that. That's the only thing I knew. That was you probably don't need a name. So I just didn't look at the names. I learned everything else about taxonomy, but not that. So I'm just like, is what we're going for? Alright. But I have a third one. I don't know where it is. Maybe it's in my drawer. I did pretty good job on speaking this time. Decent. I'm not super great, but definitely better. This one looks better than this one. Actually, you're right. Yeah, that one looks better. Yeah, that's really good. Yeah, because the one's over
because that material is actually on the lab exam that's coming up.
Yeah, that's what you have. That's what I go
I don't
I did not watch them because I know it's going on. I didn't watch it.
Okay, while you guys are getting your coats on and getting your place into a position where you can look at them, the question has come up again, what should we have on the lab quiz for Thursday? I think I will keep it. The lab quiz Thursday will cover multicellular parasites and food microbiology. And my reasoning is those topics are going to be on the laboratory exam that comes up next. Whereas this business we're doing tonight, determination of microbial numbers, use of differential and selective media, that goes on to lab exam 2. So we'll get a chance to get into that later. Yeah,
You said multicellular parasites, the food, microbiology, what was the third one?
that's it.
That was it? Oh.
That's it. We'll stop there. Because those topics are on Lab Exam 1. There is a study guide for Lab Exam 1 that is posted with the lab exam. If you looked at it earlier, if you opened it, it actually was a study guide for Lab Exam 2. Now it isn't. It's actually the right study guide. It's Lab Exam 1. What it is just a summary of each topic, and it tells you the things you need to know. You also need to look at the example Lab Exam 1. There's 2 of them that are part of the study guide. They're at the end, after all the topics that are covered as lecture questions. There's 2 examples of Laboratory Exam 1, 2 examples of Laboratory Exam 2. Please look at those, because they really give you a good idea of what to expect. Laboratory exam is 150 points. A lot of taxonomy, but remember, you are going to be given a name sheet, so don't sweat the Latin. Your spelling will be given to you. You do need to know the taxonomy, because those names are not going to be in taxonomic order, they're in alphabetical order. There's 3 columns. There's genus names, there's class names, there's phylum names, there's kingdom names, domain names, some extra names you've never heard of before, just to fill up the columns. There are no species names. Species are extra credit. Somebody asked me, if you had identified penicillium, which species can you use? The answer is any of them that are on that study guide summary. And it lists Penicillium chrysogenum, Penicillium notatum, so you can use either of those. For Aspergillus, there's Flavus, Niger, and Glaucus, I think. Any of them work. You could look up species names that I've never heard of and put them in there. Don't. That's really great. Now, then I just have to look them up. I can Google them. It's okay. Go ahead if you want to. Anyway, so that's.
Do you have practice? I'm just looking for it. Do you have a practice exam on there?
Yes, there's 2 practice exams in the study guide. They're at the end. So if you look at the study guide contents, after you get through all the topics, then there's 2 examples of Exam 1, Lab Exam 1, 2 of Lab Exam 2. Yeah.
And you didn't put all the physiological unknown plates out there, did you?
No, because some of them are still in the incubator. Those are the ones that were out were the ones that were in the incubator to begin with, and I took them out so they didn't stay in all weekend. What's in the incubator now are the ones that got in yesterday evening that are the 24 hours. So we'll get those out after a bit. Oh, first we want to look at the determination of microbial numbers. So this is your little stack of 3 plates. Some of you had Staphylococcus, some had Bacillus, some had Escherichia coli, some had Serratia marcescens. What else do you have out there? Salmonella. Anyway, so what was this about? At the beginning of each exercise, if you look at your laboratory manual, there's a section where you're supposed to look at goals. You guys are not turning in these exercises, but it's a good idea to at least think about what were we trying to do? What were our goals relative to this exercise on determination of numbers? One, I had introduced you to a number of methods that could be used to determine how many bacteria are present in a broth culture. And there were multiple different ways, and I'll go back and review those again. Population density, how many cells per milliliter, and it is always measured per milliliter. This is significant when we start trying to determine how much we've diluted a sample. So cells per mil or colony forming units per mil is what we're counting. The next thing you want to be able to do is recognize the advantages of using a viable cell count over some of the other methods that we're looking at. Then you actually got to perform a viable cell count, and the data from that is what we're going to look at tonight. So we should be able to look at that data and actually obtain the density of the population that was being investigated. In other words, how many cells per mil were in the original tube that I gave you when we started this exercise. On the other hand, do you think there were possibilities of error in this exercise? Oh yeah. There were so many pipette bulbs that had bacteria sucked into them, and people found out that trying to use 1 mil pipettes accurately is challenging. So there's some possibility of error. And usually what happens if you're working in a laboratory and you do something that's not quite right, what you are supposed to do is record what you did, because sometimes that little variation may give you better results than something that was done previously. This is why keeping track, keeping notes is so important. Anyway, so some of the methods that I had mentioned for determining the number of cells present in a sample of liquid broth medium. One is a direct microscopic count, where you have a slide that has a grid etched into it. You count the number of cells per square. You count not every square on that slide, but you count a certain number of them, and it will give you an approximation of how many cells are present. Another method is turbidimetric, where you're looking at the degree of turbidity, how much light is prevented from passing through the broth, and that's going to be influenced by how many cells are in there. So there are devices, spectrophotometers will do that for you. A third method is to use an electronic counting device, where the cells go through an aperture and each cell that goes through gets counted. All of those methods will count everything that's present. Is that what you necessarily want? No, because sometimes you want to know how many of those cells are actually alive. We've seen the growth curve, lag phase, exponential growth phase, stationary phase, exponential death phase. The cells might be present but dead, and that's going to have a much greater impact. I mean, you want to know if the cells are alive or not. So the alternative method is a viable cell count, and that's what we did. There's lots of reasons why you want to know how many cells are present that are actually living cells, especially if they're potentially pathogenic. So the advantages of using a viable cell count: it allows you to determine how many cells present in that liquid are actually living cells. You can't get the growth curve without doing a viable cell count, because you don't know how many cells are in that batch culture that are actually living cells unless you do a viable cell count. And why are viable cells the only ones that we can count? This will be a question on the lab exam, but it's lab exam 2. Well, what did we do? We took a sample, we made a serial dilution. We took samples from the serial dilution and we put them on plates. Here they are on the plates. But what's on the plate? What do you see? Colonies. Okay? The reason why are viable cells the only ones that can be counted? They are the only ones that will grow and form colonies you can see to count. If they're viable and they're in contact with the medium, they're going to grow and they're going to form a colony that you can see. If they're not viable cells, you can't see them because you can't see cells. So sometimes students will tell me, viable cells are the only ones we can see. No, you can't. You cannot see cells. You can only see colonies. So you have to tell me, viable cells are the only ones that can grow, reproduce themselves, form colonies that are visible to the naked eye. You have to be able to see colonies in order to count. Okay, so again, why do we care? Well, maybe the sample you're looking at was some kind of an additive that was going into peanut butter. This is not in the news currently, but it was not very long ago, where an additive that was going into peanut butter had salmonella in it. Had somebody been checking to see how many viable cells were in there, and had they recognized these are salmonella and they're alive, you never put salmonella into food. Never, ever, ever. Salmonella is a gastrointestinal pathogen. They will make you sick. So they're not supposed to be in there. Maybe you're looking at milk coming from a specific dairy. If you're running the bottling company, you want to know if somebody's sending you milk that has a high level of viable cells that are potentially pathogenic, something like Escherichia coli. Like, no, those guys aren't supposed to be in there. And you might reject that milk. And next time, just tell them, never mind, keep it. Or how about if you're looking at a sample of cerebral spinal fluid? Should there be viable bacteria in there? No. It's indicative of a disease situation, and again, because we know that bacteria reproduce exponentially, even a few cells can be dangerous, because a few cells will double in number very quickly. Their generation time is relatively short. Okay, so the next part of this exercise is what is a serial dilution? This again is something that can come up as a question on a lab exam. I can ask you to define serial dilution, or I can ask you to put the term in, fill in the blank. A serial dilution, it's diluting a substance to a known extent by using a series of containers where the volume of liquid in the containers is known and the amount of liquid you transfer from one container to the next is known. So you're able to determine the degree of dilution. It's not arbitrary. You can calculate the degree of dilution. In this, the next question is, why was a serial dilution necessary? Well, just take a look at your plates. The least amount of dilution we have is 10 to the minus 5. Because we didn't put the 10 to the minus 3 sample on a plate. We just used it as a dilution step. So if you look at that 10 to the minus 5 plate, then ask yourself, what would happen if you didn't dilute, you didn't set this bottle situation up, but you just took the broth sample that I gave you and you put 1 milliliter of that on a plate? What are you going to see? It's going to just be covered, and actually it will not form colonies at all. If you took a milliliter of that culture and put it on a plate, you're going to get what we call a lawn. It's going to be solid growth, no colonies. It will just be a solid layer of growth. That's called a lawn culture, not a grass culture, but a lawn culture. So why did we have to dilute it? Well, because we want to see isolated colonies. And we do a serial dilution so that we know to what extent we diluted that sample. We know that dilution is necessary to get isolated colonies. Usually you dilute with your lute, where you streak a series of quadrants, or you streak several different times. You're diluting the culture. We want isolated colonies so we can see if that culture is pure. In this case, we're diluting to a known extent. We know exactly how much we diluted, whereas if you streak the plate, you don't. Hopefully you have a plate that looks something like that. So we want to know how many cells were present in our original broth culture. The only way we can do that is to know the extent to which we diluted the culture. The exercise is set up such that the bottles are, you know what the dilutions are, because it tells you in the lab manual. But you still need to know how to determine the degree of dilution. You have to know the degree of dilution because what you're going to do is count the number of colonies on a plate and multiply times the dilution factor. So you have to know what it is. By the way, when we count colonies, a statistically acceptable number is between 20 and 200. If you have less than 20 colonies, you're not supposed to count. If you have more than 200, you're not supposed to count. However, we only do this exercise once. So if on your set of 3 plates, the only one that's countable has somewhat less than 20, go ahead and count it. Because otherwise you may not be able to count anything. Or if it has somewhat less than 200, if you really have a lot of colonies, you can divide the plate in half or divide the plate in quarters, count 1/4 and then multiply by 4. You don't have to count every single thing on that plate. Okay, so let's take a look at the degree of dilution. What I'm going to tell you here is a simplistic way of determining how to figure out your degree of dilution. And the most important thing about this is that you want to get your dilution factors into exponents as quickly as possible. Because we have individual dilution factors. Anytime you do a serial dilution, you have a series of containers. Each container has an individual dilution factor. But ultimately, you want to know the total dilution. And if you can get the numbers into an exponent form to determine the total dilution, all you do is add the exponents. So you really want to get it in an exponent form. Hope that makes sense. So how do we determine this? Well, we have our bottles. Like I said, initially, the exercise is set up. It just says, label your bottles like that. For a lab exam, they won't necessarily be. So you need to know how to do this. Our first dilution, we were supposed to transfer 0.1 mils into a bottle that contained 99.9 mils. The numbers, the volume that's in the bottle, I'm changing the number so that we always get 100 mils when we've transferred. Some exercises tell you that if you transfer 0.1 mil into your bottle, then you're supposed to add another 0.9 to make sure that it's actually 100 mils. We don't know if it's 100 mils anyway. You put 100 mils in a graduated cylinder, then you dump it into the container. There's water in that cylinder. Anyway, assuming it's 99.9, that's going into the first bottle. We want to know, is this a 1 in 10 dilution, a 1 in 100 dilution, or a 1 in 1,000 dilution? And the reason we want to know is because we can convert those into exponents very easily. 1 in 10 is 10 to the minus 1. 1 in 100 is 10 to the minus 2. 1 in 1,000 is 10 to the minus 3. And you want to get them in exponent form. The total volume we get when we transfer 1/10 of a mil into 99.9, the total volume is 100. But since the amount we transferred was 0.1 instead of 1, the dilution is 1 in 1,000 rather than 1 in 100. One way to look at this, again, I'm trying to give you a simplistic view. You always want to know what, again, if you can get it into exponent form, 1/10 is 10 to the minus 1. 1 over 100, 1/100 is 10 to the minus 2. And 1/1000 is 10 to the minus 3. If you transfer 0.1 mil, you put that into a volume that is 100 mil total. You always want to know how many organisms or how many colony forming units are in 1 mil. So we want to change this to 1 mil. A simplistic way to deal with this is move the decimal point over so that that becomes 1 mil. And if you do that, you move that decimal point over, you have to move this decimal point over, and it becomes 1 in 1,000. 1 in 1,000 is 10 to the minus 3. It's a very simplistic way to deal with this, but it works. And so many people get to this point when they're doing this exercise and they can't calculate the individual dilution factor, and it's very simple. You can look at it and say to yourself, if you're transferring 0.1 mil, you put that into a bottle and you've got 100 mils, you know that isn't 1 in 100 because it wasn't 1. So what do you do with it? Move the decimal point over. Move both decimal points, and it becomes 10 to the minus 3, which was the original dilution. Again, 1/10, 1/100, 1/1000, but you need it in exponent form. You need to know what are the exponents. Make sense? Okay. So the individual dilution factor and the total dilution factor on the first bottle are the same, because you've only got that one step. It's 10 to the minus 3. Then we go to the next bottle. And for our next bottle, we were transferring, well, liquid from the first bottle into the second, but the amount we transfer is 1 mil. So now we're going to say that our second bottle contained 99 to start with, so when you transfer 1 in there, it's 1 added to 99, so your dilution is what? Is it 1/10, 1 in 100, 1 in 1,000? It's 1 in 100. You put 1 into 99, it's now 100, so it's a 1 in 100 dilution, which in exponents is 10 to the minus 2. So our individual dilution factor is 10 to the minus 2, 1 in 100. But what is our total dilution factor? To get the total, you add your exponents. The first exponents were 10 to the minus 3. The next one is 10 to the minus 2. You add 3 and 2 together, what do you get? 5. So the total dilution factor on bottle number 3 is 10 to the minus 5, which you had already marked on there. But again, you need to know how to figure it out. Just add the exponents. So you need to get your individual dilution factor in exponent form. Then the last step, we transferred 1 mil from the second bottle into the third. Again, it's 1 into 99, which is a 1 in 100 dilution. Expressed in scientific notation, it's 10 to the minus 2. So the individual dilution factor on bottle number 3 is 10 to the minus 2. What's the total dilution factor? It's 10 to the minus 7, because you have 3, 4, 5, 6, 7. Just add your exponents. 3 plus 2 is 5, plus 2 is 7. Total dilution factor on the third bottle then is 10 to the minus 7. So they were labeled that way, but for a lab exam, I could change this up. What if I said, okay, in our third bottle, we're again transferring 0.1 mil into 99.9. Then what's the total dilution factor going to be? Yeah, it's got to be 10 to the minus 8 because that last one is another 10 to the minus 3. So it's 3 plus 2 plus 3, which is 8. 10 to the minus 8. So you need to be able to calculate the total dilution, which means you have to be able to calculate the individual dilution. So let's look at our data. If you're lucky, you have data that looks something like this, where you have very clearly a nice countable number of colonies. You're supposed to only count one plate, the plate that has between 20 and 200 colonies on it. So in this case, it would be this plate. This plate has 39 colonies on it. So this would be our countable plate. The other two samples, we say that those colonies are too numerous to count, or TNTC. Too numerous to count because we don't want to go over 200. Now, as I said, because this is the only time we do this exercise, if the only thing you have to count has way more than 200 colonies, you have to count it because we don't have anything else. Ideally, you have nicely separated colonies, you can count them. Now, as I said, the last plate there had 39 colonies on it. So what we are supposed to do to determine how many cells are present in the original sample, you count your colonies, and you multiply times the dilution factor expressed as a positive number. All you have to do is think about this logically. You cannot have a negative number of colonies. They're real. So you have to change the exponent. And so then the question is, can you write 39 times 10 to the 7th and have that be correct? Why not? Because you can't have 39. In exponential numbers, you can't, or in scientific notation, you can't have a number bigger than 10. I mean, yeah. If you do that, then you have to move the decimal point. Anyway, so 39, we have to change that to 3.9 to make it a small enough number that this will work. And when you move the decimal point over to make it 3.9, the exponent goes up. Instead of being 10 to the minus 7, excuse me, back up. Instead of being 10 to the 7, it's going to be 3.9 times 10 to the 8. That's the number of cells in the original sample. 10 to the 8, does anybody know what that is? Well, 10 to the 6 is a million. So 10 to the 8 is 100 million cells per milliliter. That's a lot. But it's not M concentration. Okay, so here's another data set, and this might look more like what you have, where you don't have a nice uniform distribution of colonies on that last plate. If you don't, there are some options. So let's say we know that we need to count our 10 to the minus 7 plate, but we're not sure what's going on here in the middle because we just have a mass here. So what we can do is say, well, let's just count this 1/3 and then multiply by 3. So we can do that. We can say, well, let's count that much of the plate and ignore the other part of the plate. As I said, you can divide the plate in quarters, divide the plate in eighths. Just don't forget to multiply by, whatever fraction you decide. Now, this is just asking, do you think any of these cultures will have reached M concentration? M concentration is 10 to the 9 cells per mil. The setup for this wasn't necessarily ideal because what I did is just pick a sample of cells off of a plate with a loop and put them in the tubes. So are the tubes, do they all have the same initial number of inoculation? Yeah,
This
not
one's
so much.
supposed to have the most, though.
But
This one's
anyway,
supposed to have
so
the
we're going to
least.
find out. These guys did have about 24 hours to grow. In order to find out, what are you going to do? You're going to count the number of colonies on a plate, that has between 20 and 200 colonies on it. You're gonna multiply that number of colonies times the dilution factor, whatever it is. And then what I want you to do is put that number on the board. And actually, I'd like you to put the numbers on the board in incorrect scientific notation first, and then we'll fix them. So just in case there are people that don't know what we have to do to make this right. So I'll put the organism categories up here.
Well, ours is like, it's like, is that the good example?
Ours looks like. Show
The
them
thing
what
is,
ours
we did
looks
it
like.
right.
Look what
How
ours
do you
looks
The thing is, we
Literally
did it
just
right.
hand
I
them
don't
this
know.
one. They're all the same. What'd you guys do? Did we not treat ours?
Okay. Again, what I want you guys to do is count the number of colonies. Let's say you count the number of colonies on a plate, and you see that you have 50 colonies, and the plate is the 10 to the minus 6. So it's 50 times 10 to the 6. Leave it wrong, and then we'll fix it as a group, okay? And please use the white chalk up here, don't use the colored chalk.
We did everything we were supposed to do.
We should have 2 inputs for each organism type.
I don't want to do the cells too. She's bullshit. I can do it like that. Hey, sorry, are you serious? Asshole.
Did we do it right?
We did everything right. We followed procedure. Yeah? I fucking hate this show.
Alright, let me see it. We need to ask them, be like, hey, this is what it looks like. When they count the dots or what? Are we only supposed to look at one?
The best one is like, I don't know. So annoying, dude.
She said count this third of it and then times it by 10%.
What'd she say? Did she say anything else?
No, I asked her if it was something wrong and she said no.
We have a lot to do tonight, so please don't take all night doing this.
I saw their results and didn't look like
Just
ours.
count it. Just draw a line and count it.
People,
No, you count on whichever one you can. So, but look for an area where you can count and count those. Okay. And if it's a third of the plate, you multiply by 3 before you.
That might be a little better.
Yeah, that's fine. It doesn't have to be the 10 to the 7.
So is this one?
I'll say 32.
Did you multiply by 3?
No.
You have to.
I know, I'm
You got 32 and a third?
It's gonna be like a little bit
That's the
long.
only one that's counted.
Well, we could probably count like half of it, actually.
Count a third.
She said count a third. So I think she doesn't want us to include half because it's such a mass thing. She doesn't want us to go where the masses are. She wants us to go where there's like colonies.
I counted 22.
You need 22, I don't really fucking care.
I just got 32.
I counted it.
Yeah, but you can't like count those either, the ones that are going into the mass.
Oh, I counted
Let me
those.
see it.
I counted anything that looked like a colony.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 22. Definitely don't have that many.
Huh?
Tell me what you get.
There's like a cluster of them there.
Kind of what you get so we can just average it.
You did a third grade?
Yeah, she did a third. Did you think my painting sucked because you couldn't see it? The first one. It was all blue, and then I realized, and you're like, that painting sucks. I was like, wait, can you see it? Because it's literally all blue.
It's not a negative number, but it's a super green number.
I got 22. 22 times 3, so it's 66.
In real life, all the things we started with, you're always gonna have. You're never gonna have 10 billion cells and have one of them be
You have to multiply by 3.
You can't have one bacteria.
one bacteria.
Right, 67. I know, fuck. 6 times 10 to the 60.
It's like the mix, the wrong mix in my brain.
66 times 26.
One bacteria.
6 is my favorite number.
How did your pump come out?
That's why I was explaining why you have
I don't know what happened with mine.
What?
How'd your pumps come out? So,
10 billion cells, you would not want them to be bacteria. You have, they'd all be bacteria, you know.
Okay, where are other numbers? The numbers up there, we should have 2 for each category.
To take us to the student center, sir.
No.
They had said to take
oh
us to
yeah.
the Coonies on
Fuck yeah.
Tuesday.
Fuck yeah, Plat B. Fuck yeah, it's going down.
Completely.
Can I give it to the 3 of us? Fuck yeah.
That's easy.
Thanks, Sunny, for making me blow my nose and think about you.
Where's the
No.
Why can't I say
Yeah, so if you counted 1 half, and you had 8 on there, you double that. And then like
This plane comes in on time. Jay, you gotta give me your flight info, bro. Jay.
You can take your anything.
Jay. Hey, Jay.
I want to study for a while.
Hey, Jay. Hey, Jay.
Or lecture.
do we not have any serration?
Hey, Jay.
Serration, please.
You gotta
Well,
send
then
me
are you
your
gonna come to the Coonies? Because we have an airport.
You're getting picked up at 2?
I did back. I did too. Oh.
What? What?
Send me your flight info.
Well, like I said, there were a lot of possibilities for error with this exercise. Go ahead and put that there. Is it half with serration? Wait,
What is wrong?
Oh.
there can't be 3 serration.
Whoa.
You don't know about
Huh.
Okay, somebody who put their number on salmonella doesn't have salmonella, because we only had 2 salmonella, and there's 2 serration. Oh, you guys put your
She said to put it up there wrong.
Well, not with a minus. Oh, I like that.
Oh, yes. I tried to flip them really fast.
Okay, so now what we're going to do is convert this to correct scientific notation. So when we look at these numbers, 28 times 10 to the 7, that doesn't fly. So what we have to do is put a decimal point here, and it becomes 2.8. When we move the decimal point over one place to the left, the exponent goes up by 1. So it becomes 2.8 times 10 to the 8, which again is not quite M concentration, but it's okay. This one is 6.2 times 10 to the 8. So those are, well, kind of similar. At least they're in the same same exponent. This is 9.6 times 10 to the 8. This is 7.0 times 10 to the 8. Most important is recognize when you put the decimal point in there, you're moving it from here to here. You're moving it one place to the left, the exponent goes up. People want to change it to 10 to the 6th. No, it's got to go up. So this one, this is 1.3 times 10 to the 7. This is 6.6 times 10 to the 7. The bacillus organisms are larger than the Escherichia coli or Salmonella. That might be a possible explanation. We tend to get lower numbers. This is Bacillus cereus, and the cells are at least twice as large as the Salmonella and E. coli. So maybe that's why those numbers are lower. We don't really know. Anyway, this is 2.0 times 10 to the 8. Now we have a 3-digit number. We have to move the decimal point over 2 places. This becomes 1.4. And because this is less than 5, it's going to go away. But our exponent is 10 to the 8. We moved it over twice. So we're fairly consistent with these guys. Then we get to our last one. So on this one, this is 10 to the 6th. We have to move the decimal point over 3 places, which is going to make this 2.0 times 10 to the 9th. And this one, we have to move it over 2 places, and it becomes 1.4 times 10 to the 9th. So of the various cultures we were working with, Serratia marcescens is the only one that reached M concentration according to this data. So 10 to the 9th is 1 billion cells per milliliter. And again, if you were writing this on an exam, you wouldn't just put these numbers, you have to put the units. This is colony forming units per mil. They're all colony forming units per mil. Yeah.
So M concentration is any number times 10 to the 9th?
Yes.
So
M concentration is 10 to the 9th cells per mil. Any number. Yes. Is that really valid? It's like cells per mil. Cells or colony forming units per mil.
so
Yeah, that's M concentration for bacteria, but that's, in a batch culture. So how does it relate to our population? It suggests to me we're dangerously close to carrying capacity. And people predict that the human population is going to keep going and keep going. Does anybody know where we are? 7.5 billion. Scary numbers. Okay, so again, you need to know, you count the number of colonies. Remember to drop the minus sign on your exponent. You can't leave it a negative number. You count the number of colonies, multiply times the dilution factor, and then put it in correct scientific notation. Okay, so that's really fairly easy. The hardest part of that exercise is calculating the degree of dilution, each individual dilution. So you need to practice and make sure that you feel comfortable doing that. Figuring out, is it 1 in 10, 1 in 100, 1 in 1,000? Okay? Now, the plates that we want to look at next are still in the incubator. Please pull out your stack of replica plates. You can leave the rest of those in there for a few moments.
Shots coming from the 12th floor.
Wasn't the 12th floor?
The cops say that it was put on the 34th floor, but you can see the shots coming from the 12th
Fucking story.
Yeah, those type of guns should not be.
No, the guy, they said that the guy was he modified his own gun. You can't buy it. He modified the gun that he was shooting. Like those guns, you can't buy it automatic rifle.
There's like more gun control saying people who want those guns are gonna get them through the black market.
No, the guy who bought the guns, all the guns he bought were illegal. He was running through it. He's just a fucking psychopath. How do we do?
I don't know, I can only see these ones in the top one. Doesn't seem too bad.
Right, and then measure it multiply twice.
Yeah, but I saw my physiological unknown in there. It was pink. It's really pretty.
Right.
That way you're not multiplying.
It's like, that's one of those math things, you gotta do the right thing, right
I like
order.
all the colors.
So I'll go and put some
Don't do that, man.
Okay, so once you get your replica plates, you're gonna have to take the tape off of them and put them into a position so you can actually look at them.
Let me see the tape.
You know what you're gonna do.
You have a contaminant?
I don't know what to tell you. This nigga do shit. Doesn't do shit. Huh?
Everybody's like blaming Trump for it, but it's like supposed to be shit. It's some really great information. Trying to
Did you
sabotage.
look at the line?
So again, you do not necessarily have to get your physiological unknowns out at this time. What I want you to have is that stack of plates. So you have a stack of plates, and on top is the replica master, or somewhere in there is replica master. So this is our use of selective and differential media replica plating exercise. Once again, we want to know what is it we're trying to do. So goals for this exercise. You're going to become familiar with some new media categories. Our initial media categories that we looked at were complex and defined. For this exercise, the new media categories were selective and differential. Turns out the media categories are not exclusive, so the media that we're working with tonight are selective and differential and complex. you were supposed to assemble a replica plating block and then use it to transfer multiple colonies. We had 5 different types of bacteria. You were supposed to transfer them onto a series of plates. So, again, the media used in this exercise are all selective, differential, and complex. What we're going to be doing now is taking a look at them. You need to know what makes the medium selective. So each medium type has some aspect that makes it selective. The other thing you need to know is what is the basis for them being differential? In other words, how are they differential? What do they do that causes you to be able to tell cells apart? So selective medium. A selective medium is one that allows only certain types of organisms to grow and inhibits the growth of others. A differential medium allows multiple different types of organisms to grow, but causes them to look different. So we're going to see a series of media and see how they do this. Before we start that though, you're supposed to look at the last plate in the replica series. This was a nutrient auger plate, and this is a control plate. Does it have 5 sets of colonies on it? So your replica master had 5 sets of colonies. Your control plate should also have 5 sets of colonies. What does this tell you? They were all transferred. This is just a logic thing. If you had a master plate and you put that pattern on your velvet, and then you did McConkey's, EMV, T7, mannitol salt, and then nutrient auger at the end, that last plate has all 5 colony types. That means you transferred all 5 colony types to all the other media, assuming that you did the process the same way, where you made sure that the velvet hairs touch the plate surface in each case. Okay, so again, this is a positive control. If you see colonies on there, that's what the master looked like. The control looks a little different, but you can still see that you've got all 5 colony types on there. So that tells you you did this process correctly. And it tells you that all 5 cultures should have been transferred to every single plate in between. Yeah, that's what the growth on that plate tells us. Okay, so the first selective and differential medium we're going to look at is mannitol salt auger. This is the red plate, kind of an orangey red as opposed to pinkish. This is a medium that contains 7.5% sodium chloride. It's in the name, mannitol salt. But 7.5% is very high salt. What kind of organisms is this going to be selective for? Halophiles. So halophiles, organisms that like a high salt content. So there's the medium. The halophiles that typically are grown on mannitol salt auger are Staphylococcus. Staphylococcus are organisms that normally live on the surface of human skin. They also live in our nasal passages. Have you ever tasted the surface of your skin on a hot day after being working outside for a while, several hours? It's salty because perspiration contains salt, and the water evaporates, and the salt builds up, and pretty soon the surface of your skin is very salty. How about the inside of your nasal passage? Never mind. That's salty too, because you're producing secretions, the water evaporates, and the salt builds up. So Staphylococcus tend to be halophiles. So they're going to be growing on this. We're hoping to select for them. This medium is differential on the basis of mannitol fermentation. So mannitol is the carbohydrate present. Again, it's in the name, mannitol salt auger. So mannitol is the carbohydrate that's in there. There's a pH indicator. The pH indicator is phenol red. If the organisms can ferment mannitol, they will produce acid, and it will cause the pH indicator to change color. It turns yellow. If they can't produce acid, but they actually produce alkaline end products, the phenol red will turn more of a pinky red, kind of a hot pink. I doubt if you're going to see that on this plate. Here we see 2 cultures on the mannitol salt plate. Now, to read this correctly, don't look through the bottom of the plate. Turn it so that you're looking through the lid. You can take the lid off if it's steaming, you can't see through it. You should have yellow colonies at the top and white colonies at the bottom. The yellow colonies are Staphylococcus aureus. That is the colonies surrounded by a yellow zone. They ferment mannitol quite nicely. Staphylococcus epidermidis, which are the ones at the bottom, do not. Do you guys see anything else growing on that plate? Okay, so that's something you should be paying attention to. This medium is pretty good selective, but we've got culture in the middle here, and we've got a hint of something growing over here on one side. So it is selective, but it's not completely selective. We're getting a little bit of something else growing on that.
You said if it ferments, mannitol will be yellow?
Yes, because it's going to produce acid, and acid will cause the phenol red indicator to turn yellow. Yep. Okay, so hopefully some of you see exactly what I'm showing you on the screen, and if not, at least you get to see it on the screen. The next three, Turgitol 7, McConkey's, and EMB, are all similar in terms of their being selective. So these are all selective because they have chemicals in them. Chemicals that tend to inhibit the growth of something. Some of the chemicals are basic dyes, such as eosin and methylene blue. Some of the chemicals are something else. But which general type of bacteria is more sensitive to chemicals? Gram-positive or gram-negative? Gram-positive, because remember gram-negatives, their wall has an outer membrane, and that outer membrane tends to inhibit chemicals coming through. So gram-negatives are less sensitive to chemicals. Consequently, these media are selective for gram-negative. Gram-negatives will grow, gram-positives typically won't. And then we can look at these individually because they have different pH indicators associated with them. So Turgitol 7 medium contains Turgitol 7. I'm not sure what it is, but it does inhibit the growth of gram-positives. So it's selective for gram-negatives. It's this kind of light green color. For a while, this medium, the company that was producing it was giving us a medium type that was very dark. And this semester, they opened a brand new bottle, and it's back to being the color it's supposed to be, which is that kind of pale green. I'm happy. Anyway, the pH indicator in this medium is bromthiamyl blue. Bromthiamyl blue is green when it's neutral. It turns yellow if it's acidic, and it turns blue if it's alkaline. And on this light-colored medium, you can see it quite nicely. This medium contains the disaccharide lactose. So if the organisms can ferment lactose, they're going to produce acid, and it will turn the medium yellow. If they can't ferment lactose, and the medium turns blue, you might be looking at this and going, why do they turn blue? All of these media contain peptone. Peptone is protein breakdown products, so you've got some amino acids, and bacteria can catabolize amino acids and produce substances called amines, and amines are alcohols. So we actually end up getting two colors. This medium shows growth on the left and on the right. On the left, we see colonies that are surrounded by a yellow zone, and on the right, we see colonies surrounded by a blue zone. Sometimes it's green down the middle. Sometimes you kind of lose the green. Okay? So, Escherichia coli ferments lactose. Salmonella does not. Which side is which? Escherichia coli is on the left. So the colonies that are surrounded by the yellow zone, that's Escherichia coli. Salmonella colorsus cannot ferment lactose, but it can degrade the amino acids in the peptone, and it actually turns the medium blue. Notice Staphylococcus doesn't grow in this medium. So our Turgitol 7 medium is a pretty good selective medium. We rarely get any growth top and bottom. There's some stray colonies out there, but I think they fell off the velvet and they just randomly dropped. There's no growth top, bottom, or middle. So Turgitol 7 is a pretty good selective medium. It's selective for gram-negative and differential on the basis of lactose fermentation. McConkey's auger. This particular McConkey's auger that we use contains crystal violet, which is another basic dye. Crystal violet will tend to inhibit the growth of gram-positive bacteria, so McConkey's is also selective for gram-negatives. And it also contains lactose. The pH indicator in McConkey's auger is neutral red. This is the only place where we see that particular pH indicator. if the organisms can catabolize lactose, if they can ferment lactose and produce acid, they will accumulate neutral red in the colonies and sometimes around the colonies. And it's the crystal violet in the medium that causes the neutral red to kind of have a pink color. Otherwise, it's not supposed to look like that, but it's definitely pink. So again, growth on McConkeys is on the left and on the right. On the left is Escherichia coli. Those colonies can ferment lactose. On the right is Salmonella. Another thing about the Salmonella colonies, they tend to be somewhat less opaque. They're a little more translucent. And they have actually changed the color of the McConkeys from pink to a kind of a yellowish tan. So again, they are producing alkaline end products, breaking down the peptone in there. And we see that this McConkeys medium is, again, pretty selective. We don't have growth top and bottom. We don't have growth in the middle. That's why we used natural salt agar, McConkeys agar, and T7 for the physiological unknowns. We did not use eosin methylene blue, which I'll show you in a moment. The eosin methylene blue, again, contains basic dyes, so it's going to tend to inhibit the growth of gram-positives. It doesn't have a pH indicator in it. It still has lactose in it. It's this dark, kind of burgundy-colored medium. The gram-negatives can grow on here better than gram-positives because of the basic dyes. But apparently, if the organisms grow and ferment lactose, they somehow accumulate the dye on their colony surface and they produce colonies that are iridescent. Iridescence is an optical character. It's light reflected off the surface. So they're supposed to be iridescent green, and the colonies that are Escherichia coli. This is much better when you see it actually on the plate than when you see it in a picture. And this, again, this color is transient. That iridescent green shows up pretty well, even now. You can kind of see it, but you guys don't have light. You can only kind of sort of see it. And this medium, this one has no growth top, bottom, or middle.
Oh, see?
Mine had growth on all over the place. Does everybody see the iridescent green?
Yes.
Okay. So again, that's reflected light. That's iridescence. It's an optical character. So Escherichia coli is on the left, Salmonella is on the right. This particular plate, we had a little bit of growth from the staff and also from this colony in the middle. But this one didn't. Did anybody else see growth in other places with the EMB? Or is it just right, left? Yeah, okay. So it tends not to be quite as selective. So Staphylococcus grew on my plate and the culture in the middle. So about that culture in the middle, do you think they're gram-positive or gram-negative? They should be gram-positive because they didn't grow on T7, they didn't grow on McConkeys, they didn't grow well on EMB. Anybody recognize them? The ones in the middle. Notice they're kind of spready. Surface texture is kind of textured, like frosted glass.
Bacillus?
Bacillus. Yeah, that's Bacillus cereus. That was from our mixed B plate, the first plates that we did. Yep, so those are Bacillus cereus. They are gram-positive. Now, the process, the replica plating, is supposed to save time and save media. Because instead of taking each organism type and streaking it one at a time onto a medium, you got to move all 5 types at once. Was it easy? Yeah, way easy. So it saves time, it saves money, it's quick, it's easy. Yeah, it's a good thing to do. Not difficult at all. Now, I want to talk about some of the other media that were used for the physiological unknown 1. These weren't used in the replicating exercise. They're just something we have out for physiological unknown 1. Some of you were supposed to streak Mueller-Hinton agar. Mueller-Hinton agar is non-selective, but it can be differential because it will tend to promote the growth of colonies that produce pigment And if they can produce pigment, a lot of times the pigment will show on Mueller-Hinton where it doesn't show on nutrient. So in this picture, you can see the colonies that are showing up, most of the growth is on the left. They're producing a water-soluble pigment. It's green. It's causing the color change in the medium. Sometimes the differences on Mueller-Hinton agar can be very subtle. So here we see two different colony types, but you have to look at it closely. You can see that these guys are smaller, these are bigger, these are, actually you can't see from back there, but these are little bitty convex colonies where these larger ones are raised or almost flat. So look at the very subtle differences. Remember, when you're looking at colonies, we have to look at form, elevation, margin, surface texture, optical character, size, color, all kinds of things. She wants to look carefully. Another medium type that some people got to use was nutrient agar. Again, it's not selective, but it can be differential just because colonies can look different. So here we see bacillus and something else. Bacillus colonies are kind of rough on the surface and irregular. The other little colonies are circular, entire, more white. Blood agar, also not selective. Blood agar is an enriched medium, not enrichment, but an enriched medium. It contains 5% sheep blood. Here we see some colonies on blood agar. Can you tell which ones are white? Now, on blood agar, again, it's not selective. Pretty much everything we have will grow on blood agar. The fastidious organisms, which will only grow on blood agar, tend to produce colonies that are pretty small. So those of you that streak blood agar plates, you may have two different things on there. The fastidious colonies should be smaller, and usually they're white. Kind of hard to tell, but on this plate, these colonies that are large and kind of off-white, kind of tannish gray, those are not fastidious. This little white one is. And the longer you leave them on there, the non-fastidious ones will get, the colonies get quite a lot bigger. Blood agar can also be differential on the basis of hemolysis reactions, which I'll talk about later on. There's alpha, beta, and gamma hemolysis on blood agar. And then the media that I don't have pictures of, brain-hard agar. Brain-hard agar is also an enriched medium. Actually, I want to put some information on the board, just kind of a summary. Things that we know about the media that we're going to be using tonight. And about physiological unknowns. Okay, so we're switching over. Now we're going to be working with physiological unknown 1. Your goal tonight is to establish 2 pure cultures. So how many physiological unknowns do you think you have? 2. Everybody has 2. Okay? If you have something gram-positive, of information for you. Everybody has two. If you have anything gram-positive is going to be accompanied by 1 gram-negative fermentative culture. But not everybody has something gram-positive. How are you going to find out if you have something gram-positive? You're going to do a gram stain, right? So a gram stain is going to be required. Remember that I already said I would like you to do gram stain from the sauerkraut. I would like you to do gram stain from the buttermilk yogurt plates. There's 2 unknowns. I want you to gram stain both of them. I'd like you to use 1 piece of glass. It's doable. You just have to make your smear small. Okay, some other things we need to know. Some of you can have 2 gram negatives. Some people have 2 gram negatives. If you have 2 gram negatives, 1 is fermentative. Everybody has 1 gram negative fermentative culture. The tricky part is determining what else you might have. So if 1 is fermentative, the other can be respiratory or fastidious. So you might look at this and say, how are we going to know if they're respiratory or they're fastidious? Let's start with the easy one. How do you know if your culture is fastidious?
Small. It's small
on
specific media, enriched media.
It has to be grown on enriched media. So fastidious will only grow on enriched medium. Our enriched media are blood or brain heart. Last semester, people started calling BHA blood heart auger. We don't have blood heart auger, we have brain heart auger. But both brain heart auger and blood auger are enriched. So if you have one or the other of these, you know you have 1 fastidious culture. That means your other culture is gram-negative fermentative. If you have 2 gram-negatives and 1 is fermentative and 1 is respiratory, there's a test that you're supposed to set up tonight called the oxidation fermentation test. Or OF test. This requires that you inoculate 2 tubes with both of the different organism types, which is a little bit tricky because you have to get them apart before you're supposed to inoculate anything. If you think you have 2 gram-negatives and 1 is fermentative and 1 is respiratory, see me about doing an OF test. Okay, now, the other thing is to remember what we know about our media. T7 and McConkey's selective. Selected for what? Selective for gram-negative. So if you have growth on T7 or McConkey's, you should have gram-negative culture on there. Those are pretty good selective media. You should not have random gram-positives growing on there. Mannitol salt, on the other hand, This is selective. For halophiles, it will grow staph, but it might grow something else. We saw on our mannitol salt plates, there were some other things that might grow on there. And we have some gram-negatives that are halophiles. Nevertheless, these are our selective and differential media. And what you're supposed to do tonight is, first of all, observe your plates. So you're going to observe the colonies that are on your plates. Everybody has, I think, 3 plates. Some of them are still in the incubator. Everybody has T7 or McConkie. Everybody has T7 or McConkie somewhere. And I think everybody has TSA. And then some of you have mannitol salt agar, some of you have blood agar, some of you have Hinton agar, some of you have brain heart agar. So, record morphology, please remember color. Because we are working with some media that are selective and differential, this is where the differential part comes into play. Record the color of the medium as well as the color of the colonies. And then, once you've recorded that, also record, what does this tell you? For example, color of medium, with its McConkie, and the colonies are pink. What does that tell you?
Gram.
The fact that they're growing on McConkies tells you they're gram-negative. If they're pink colonies on McConkies, it tells you they ferment lactose. I don't expect you to know everything about this right now, but there's a section in the laboratory manual that talks about these media. It tells you what the color changes mean. What's most important tonight is make sure you record it, and record it someplace where you can't lose it, because you have to do a write-up on physiological unknown 1, and part of the write-up is this, colony morphology, and what it means, what it tells you. Okay, so do a good job with this. Make sure you get all of the details. The other thing you're supposed to do tonight, like I said, is gram stain. What do you record? You record data, result, conclusion. You did this for the morphological unknown. This is just a repeat. Do it again. You just don't have the boxes to put your stuff in. But you should know how to do that. You should have a gram stain for 2 different things, because everybody has 2 things. The tricky part is if you have 3 plates, how do you know which ones are which? You may end up having to do more than 1 piece of glass. But you want to sort this out, and then you're going to restream. So, tell you what, I'm going to let you work on these guys for a little bit, because I want you to get the gram stains and sort out what's gram positive, what's gram negative, and then I'll put the information on the board telling you what to re-streak to, what medium to streak to. Okay? So your plates are, some of them in the incubator, some of them I stuck in lab drawers.
What stains is My head is exploding.
I'm curious to see what grew and what didn't. How they grew.
Do you have all three of those?
Only one more. I think it's in there. Yours too?
Yeah.
Which one?
They're really pretty, right? Well, you can keep them, but they won't take them.
The mat. And I do have pink colonies.
You can take pictures of them. Yeah. Yeah, so for your replica plates, you may take pictures of them. If there's somebody missing, you can hide them in their drawer. Otherwise, they go in the bedroom. We're finished with those.
This side, right?
Yes.
What I was gonna say.
What the heck? What did you guys do? You must not have touched the master.
That's probably
There's nothing on them?
That's what I'm saying.
Yeah, it's like what?
I'm pretty sure we did touch the master.
I don't think those media ever got inoculated.
We saw these, you're like,
That's just sad.
it's quite sad.
Where does he go?
Look at yours.
Into a coffee.
Yeah, look at somebody else's. Hopefully not.
I see your plates.
Well, maybe.
You can grow on every single one.
The thing is, you have more than one plate.
What?
I was just exhausted.
So although that looks like there's only one thing on there, what's your other medium? Yeah, so you might. This is not enriched.
Can you provide a shadow,
You might have something on the
So
flip.
like,
Hey,
You don't
maybe
feel
they do 5 minutes.
Nope, I gotta get up.
No, I had somebody like on my plate
Yeah.
and like look at with them because I didn't see it there. Yeah. Ooh, so pretty.